All right, it is noon. Welcome everybody to another Iowa Learning Farms webinar. Um, my name is Mitch Harding. I am a water outreach specialist with Iowa Learning Farms, and I'm filling in today for Dr. Matt Helmers. This week we have David Ramotowski, a PhD candidate with the Civil and Environmental Engineering Department out of the University of Iowa. And he's going to be talking to us today about using fields of opportunities to clean up forever chemicals, specifically PCBs, it sounds like. So without further ado, I'm going to pass the floor on over to David, and he's going to share a little bit about his work that he's doing at the University of Iowa.
All right. Well, thank you very much for coming and for the opportunity to talk to you today about my research. As Mitch mentioned, I'm a fifth year PhD candidate in civil and environmental engineering at the University of Iowa. And I'll be talking about how we can use our fields of opportunities in combination with bacteria to help clean up forever chemicals. And so first, for a little introduction, on the left is a structure of a chemical that we're probably all familiar with. It has a very complicated name, but we know it as DDT. And so interestingly, in the 1960s, there was a Swedish scientist who was who was looking for DDT in the environment, specifically in white-tailed eagles. And he found DDT in his chromatogram, but also he noticed a bunch of unknown peaks representing unknown chemicals. And it turns out that all of these unknown peaks were PCBs, or polychlorinated biphenyls. There are 209 different PCBs based on the number of chlorines attached and their position. And as you can see, the structure is very similar to DDT, and it is why they are able to accumulate in the environment with the same intensity. PCBs were produced from about the 1930s to the end of the 1970s, and they were primarily used in applications such as electrical transformers and lubricants, but they're also found in paints and even building materials. And so PCBs are a problem because once they get out into the environment, they tend to accumulate in soils, sediments, and also within fish and other marine animals, particularly through a process called biomagnification, where larger, more predatory fish eat smaller fish with PCBs, and then that causes the concentration to go up inside of the larger fish. And so that's why in a lot of cases we're not able to eat fish that are caught near PCB contaminated sites. And then also relevant to my research, PCBs can be emitted from sediment when they're improperly disposed up into the air where they have spread all over the world. PCBs have even been found within polar bears in the Arctic, which is thousands of miles from any known source. And they've also been found all the way at the bottom of the ocean. So they've truly spread worldwide. However, the greatest danger lies within PCBs or people living near contaminated sites where the risk of exposure is the highest. And so before starting at Iowa, I was actually a master's student in soil science at NC State, which is kind of like where my agricultural background comes from. And somewhat worryingly, within the past few years, scientists were able to link a cancer cluster at researchers who previously worked at NC State to buildings that contain PCBs because PCBs were at their greatest extent around like the 1950s, 1960s. And so buildings built during that time often contain high amounts of PCBs, and the airborne emissions from those building materials cause people to become sick. And so this is something that my research center is working to prevent, and I am working to prevent specifically PCBs being emitted from sediment to water to air. And so, how can we get rid of PCBs? So, right now, the most common methods of getting rid of PCBs or cleaning them up is to go in, take the sediment, so you dredge the sediment, and then you have to either put it into a chemical waste landfill, which is expensive and potentially dangerous if the landfill is not constructed properly, or you have to incinerate the PCBs. And this can be a problem because if the temperature is not high enough, more PCBs can actually be released into the air because it requires a very, very high temperature to break down PCBs. And so, therefore, we want to try and find a method that allows us to clean up PCBs directly at the contaminated sites, which is potentially safer and more cost effective. And so, interestingly, in the 1980s, researchers in New York were testing sediments in a contaminated chemical waste landfill, and they found a interesting bacteria called Paraburkholderia xenovorans LB400, or just LB400 for short. This bacteria was found to be able to degrade a wide range of PCBs, and because of that, it has since become the most well-known aerobic or oxygen-using bacteria in this field, and it is the bacteria that I use in my current research. LB400 is also particularly effective in degrading what we call low chlorinated PCBs. So PCBs with free chlorines or less, which is very important because these PCBs, because they are lighter, are actually more likely to be emitted into the air. And therefore, these are the types of PCBs that we really want to focus on. And so for my initial work, we wanted to take LB400 and see how it would perform in decreasing PCBs emitted from two different sediments. We took samples from Alta Vista, Virginia, which is a contaminated wastewater lagoon, and it is a freshwater system. And we also took samples from New Bedford Harbor, Massachusetts, which is a Superfund site that is well known for having among the highest, if not the highest, airborne PCB emissions in the country. This site is particularly interesting because it's an estuary, and so the bacteria, if you were to introduce them into this environment, would have to deal with a lot of stresses such as strong currents, salinity, changes in oxygen levels, and a lot of other difficulties. And so we wanted to see how LB400 would perform in these two environments. And then I'd also like to mention that both of these sites were contaminated with different mixtures of PCBs. The trade name for PCBs was Aerochlor, and they were sold in mixtures of all different types of PCBs. And so Alta Vista was contaminated primarily with Aerochlor 1248, and New Bedford was contaminated with Aerochlor 1016. And this is interesting because Aerochlor 1016 has more of those lighter, low chlorinated PCBs that we really want to try and clean up. And so I ended up growing LB400 as a liquid culture in the lab. And you can see how it looks on the right. And that's how it looks when it's finished and ready to be used. It sort of has this interesting teal color. And so I ended up preparing a microcosm experiment. So I wanted to try to approximate the real world conditions as closely as I could using a 250 milliliter Erlenmeyer flask with a 10 to 1 ratio of liquid to sediment. And most importantly, I have a piece of polyurethane foam in the neck of the flask, and this is able to absorb PCBs in air. And so that's what allows us to determine if LB400 has a significant effect on airborne PCB emissions. And so I ended up running two experiments, one with the Alta Vista sediment and one with the New Bedford sediment. And I ended up taking samples at three different times at 16 days, 35 days, and 75 days because we wanted to see how LB400 would perform over time because this is an important aspect when thinking about using bacteria in the field is that ideally you want to have it last as long as possible so that you don't have to reapply. And so we found that in both sediments, LB400 significantly decreased PCB emissions in as little as 35 days and up to 75 days, which is really exciting, and especially because it was highly effective at degrading the low chlorinated or LC PCBs, which we really want to get rid of, especially in New Bedford, given the difficulties that I mentioned. It was really interesting to see that LB400 could still be effective. And then I also extracted DNA from the sediment in both of my experiments. And I was looking specifically for the biphenyl dioxygenase subunit A or BPHA gene. And this is important because the BPHA gene catalyzes the first step of PCB degradation. And so looking at the amount of this gene can help us determine, or at least give us an idea of LB400's activity over time. And so we did notice a sharp decrease in the gene copy numbers after about 16 days. But interestingly, we did notice a recovery in the New Bedford sediment at 75 days, which I'll touch on later in this presentation. And so now that we've established that LB400 can potentially work in the field, we need to figure out a way to actually deliver it into the environment. Because if we simply took a liquid culture and added it to an environment like New Bedford, it would simply be washed away. So therefore, we need a way to protect the bacteria and to help it do its job. And so this is where the biochar comes in. My research group found that attaching LB400 to biochar can can work to decrease PCB emissions because the biochar can sort of act like a magnet and attract PCBs through absorption, while the bacteria on the surface is able to break them down. And so in this way, it's sort of like the perfect, or potentially the perfect 1-2 punch to help clean up PCBs and prevent people from getting sick. And perhaps for this audience, the most interesting aspect of this research is that my research group found that biochar made from corn kernels was actually the most effective when combined with the bacteria. It was sort of like the best habitat for the bacteria, which is interesting because biochar is sort of like a charcoal-like material, and so it can be made from a wide range of plant materials, but we found that biochar made from corn kernels actually worked the best with LB400, which was really exciting. And so taking this concept, I next wanted to figure out how much biochar to use with how much sediment, because there's a lack of consensus in the literature about exactly how much biochar you need to have a significant effect on PCBs in air. And so this is something that is very important when thinking about cost, especially when trying to scale up this treatment method. And so I prepared an experiment using the same microcosm design as before, but this time I used three different percentages of biochar to sediment, 1.5%, 5%, and 10%. And then I also had a no biochar control. And the percentages are based on the weight. So with 10 grams of sediment, 10% of that would be 1 gram, then going down to half a gram, and then zero point 0.15 grams or 0.015. And so we were really interested to see how this biochar and bacteria combo would perform over a period of 85 days in this case, because we wanted to see if the biochar could also potentially help the bacteria stay active for longer. And here you can see what the biochar looks like in the lab. We actually end up grinding the biochar before putting it into the microcosms. Sort of like the same principle when grinding coffee. You want to increase the surface area so it gives the bacteria more chances to attach to the biochar. And then on the right, you can see one of my microcosms. I've actually noticed some really interesting growth happening at the bottom of these flasks. During these experiments. And so I find that really interesting. And then getting to the results, it appeared that the 5% biochar actually performed the best because we noticed a significant decrease in low chlorinated PCBs in air after 85 days. And this is really interesting because unfortunately with 1.5% biochar, we did not see a significant effect. But with 10% biochar, we did see a noticeable decrease. But going back to the cost issue, using 10% biochar is effectively twice as much as 5%. And so we decided for our future experiments to use 5% biochar to sediment because this is sort of like the best medium between the cost and then also having a significant impact on PCBs in the air. And so, because this is the Iowa Learning Farms webinar, I wanted to do a quick set of calculations. I don't claim to be, an expert in this, but I do remember from my soil science curriculum that an acre of topsoil is considered to be around 2 million pounds. And so 5% of that would be around 100,000 pounds of biochar needed. And so if we take the average corn yield of 189 bushels per acre from the USDA, you would need just under 1,800 bushels for 100,000 pounds of corn kernels. And based on current prices, that would come out to around just over $7,000 per acre treated using the biochar. And if you're going for treating 150 acres, this would be about $1.1 million. And while this doesn't include the cost of producing the biochar itself or the bacteria, this is important because PCB remediation using the normal dredging and transport method can cost tens of millions and potentially more. And so this shows how we can sort of use Iowa's top homegrown resource to help protect public health in a more cost-effective and potentially safer way. And this is especially important in a site like New Bedford because over the decades since PCBs were banned, they still present a significant health risk to people living nearby. And almost $1 billion has been spent to clean up the sediment. And about half of that is actually taxpayer money. And so potentially, if we're able to scale up this treatment, we'll be able to save a significant amount compared to normal methods while also not needing to go in, take the sediment and remove it, which is very destructive, expensive, and potentially unsafe. And so for my most recent experiment, building upon the results where we determined that 5% biochar was the ideal amount, I wanted to see how effective this biochar would be over a long period of time in decreasing PCB emissions from sediment into the air. And so I prepared a similar experiment using the 5% biochar to sediment with 4 sampling times ranging from 11 days to 120 days. and I looked at how effective the bacteria and biochar was, along with just the biochar by itself. And then I also had a sediment-only control for reference. And so we found that, interestingly, the bacteria and biochar were highly effective at preventing PCB emissions for up to 60 days, especially for the first two time points, days 11 and 25. There was a significant decrease in PCBs in air as a result of the bacteria being added. However, at 60 days, the difference between the bacteria treatment with the biochar and just the biochar by itself was not significant. However, the difference was still very highly significant when compared to the no biochar control. And so this is very interesting because when looking back at my experiments with the liquid bacteria, we can see that the bacteria and biochar can also be significantly effective for a long period of time. And this is especially interesting because with the biochar and bacteria, the bacteria is sort of in what we would call a no growth situation because the bacteria does not like actually grow off of the biochar. And so once you add it in, it's sort of on its own. And so it was really interesting that it was able to last for so long. And then I also extracted RNA from this experiment. And so, in comparison to taking DNA samples, RNA actually shows if the gene is being expressed, which is a much more accurate way of measuring LB400 activity. And so, we noticed that at 60 days, there was a drop in LB400 activity, which correlates with the drop that we saw at 60 days in the emissions data. However, there was a recovery in activity at around 120 days. And so that sort of makes us think that because we're looking at the biphenyl dioxygenase or BPHA gene, that gene is not exclusive to LB400. It can also be present potentially in native microorganisms that could be present in New Bedford Harbor. And so potentially those native microorganisms could be responsible for this recovery that we see at 120 days. And that brings me to my future work, which is I also extracted DNA from the sediment at the time that we collected it back in 2022. And I sequenced the BPHA gene, and I found that really interestingly, the samples that we took from the shoreline of New Bedford, which I used in all of my experiments, are the most diverse when compared to LB400. And this is really interesting because it indicates that there could be native bacteria present that are better suited to the harsh conditions in New Bedford, such as the strong currents, changing oxygen levels with the tides, and other problems that could, and these bacteria could potentially, if we're able to find them, be used along with our corn kernel biochar to degrade PCBs potentially even more effectively for a longer period of time. And this is important because the longer the bacteria lasts, the more cost effective it is because you would not have to reapply potentially. And so to find these new PCB degraders, we're planning to use a relatively new method called the iChip, which consists of taking a 96 well plate, filling the wells with agar, and then putting that into the sediment from New Bedford. And we're also working on building what we call a mesocosm, which is sort of like a more advanced version of the flasks that I was using in my previous experiments. And this is really interesting because it allows us to raise and lower the water level to sort of simulate the tides at New Bedford. And that would allow us to select for bacteria that are able to survive in that type of environment. And so I'm really excited to see what we're able to find moving forward. And so with that, I really want to take the opportunity to thank one of my lab mates, now Dr. Chen Dong. She actually did the work of testing the different biochars to see, which ones work the best of our LB400. And so through her work, we were able to find that the corn kernel biochar worked the best of our bacteria. Um, she also developed a really interesting method of encapsulating the bacteria in the biochar with a type of material called sol gel. It's a glass-like material. And the idea is that this can actually let in PCBs while keeping out harmful toxins that could hurt the bacteria. And so this is another avenue that we're going to be exploring to try to further improve our treatment. And as I like to say, to use our fields of opportunities to give people the freedom to flourish in a world free from forever chemicals. And so, with that, I really appreciate all of you for coming. Feel free to contact me via email, and I'd also love to connect with you on LinkedIn. And then I'd like to also thank the National Institute of Environmental Health Sciences, which funded my research through the Iowa Superfund Research Program. So, thank you very much for coming, and I'm happy to take questions.
Yes, we've gotten lots of questions in. So continue to throw those questions into the chat box. I think we have a few other slides we want to cover, so add some questions to the chat box while we go over these last couple slides. Um, yep, so if you could go to the next slide for me. There
Sorry.
we go. Perfect, you're good. So yes, if you're watching today to get a CCA CEU credit, please email Lena Whitaker by 5 p.m. today with your name, the name you entered the webinar with, and your CCA number. Her email is alenaw@iastate.edu. So send her that email by 5 p.m. today. Next slide, please. If you have not taken our 2025 voluntary demographic survey, I encourage you to take that. We only need one response for 2025. So if you've already done it, thank you for doing that. But if you have not, I encourage you to go ahead and do so. Uh, I think next slide is our webinar for next week. Um, so yes, please tune in next week, next Wednesday at noon. We'll be hearing from Sabrina Ruiz with USDA ARS. She's going to discuss managing cover crops to achieve high biomass and improve soil health indicators. So tune in next Wednesday at noon to learn more about that. And I think we have one more, maybe. There we go. Okay. Um, Also, feel free to check out this webinar and other past webinars by looking into our archives. You can find those at iowalearningfarms.org/webinars, as well as checking out our YouTube channel, where we've got all those webinars sorted out by year and topic. So feel free to check out any webinars from our past series. All right, so we've got lots of questions that have come in over your presentation. So to kick things off, um, one question, so how did you confirm that the decrease in captured PCB was actual destruction of the PCB rather than some kind of accumulation from the bacteria?
Yeah, that's a very good question. So, so we assume that if there is a significant difference in the PCBs captured on our polyurethane foam or puff, that the difference is due to the bacteria breaking them down. One thing that we could do in the future is we can also look at the PCBs directly in the sediment and then also in the water. I did attempt to measure PCBs in the water using a type of sort of like a fiber optic cable called SPME. It's called a solid phase. Yeah, sorry, I can't remember it off the top of my head, um, but yeah, the data from that was actually really interesting because we actually noticed higher amounts of PCB on those fibers, and we think that it's because the fibers sort of like attract the PCBs, so they can sort of compete with the bacteria. And then another thing we also want to look at is if the PCBs are actually sorbing to the dead bacteria, because we assume that around like a month or two, give or take, after adding the bacteria, that it eventually dies. But one thing we don't know is that if the PCBs can still be attracted, even after that. And so that's something that we definitely want to look into in the future.
Another question that came in, are there any other environmental impacts or potential environmental impacts of introducing the bacteria or the biochar into the environment?
Yeah, I'm not totally sure about the biochar itself, but biochar is sort of inert because it's made at such a high temperature that we wouldn't expect any harmful pathogens to be growing on it. But for the bacteria, the main product of breaking down PCBs is a type of chemical called chlorobenzoates. And so moving forward, we want to actually try and measure these chlorobenzoates as well for our future experiments. Because we want to see if that could pose a threat to the microbial community and potentially to fish, other wildlife, and ultimately humans in the environment. So, yeah, that's something that we definitely want to look into in the future as well.
Another question. So some people were asking about different biochar materials. So were there any other materials that you tried for biochar, and then how did they compare as far as like costs and things go?
Yeah, that's a really interesting question. I can't remember all the different materials that we tested or that Chen tested, but we looked at reeds, corn kernels. I think she also maybe looked at just like wood charcoal. And it's interesting because, the cost is important because obviously using a more expensive material would make the treatment cost more. But then you also want to look at how the biochar performs with the bacteria. And so I remember at Chin's defense, she was explaining her process. And to sort of simplify it, when I gave the example of biochar working like a magnet to attract the PCBs, you don't want the magnet to be like so strong that it could potentially hurt the bacteria. And so you sort of want to find like the happy medium between like a really strong biochar, but then also a type of biochar that provides a great habitat for the bacteria to grow. And she noticed that the corn kernel biochar actually produced the thickest films of bacteria in comparison to the others. So, yeah, thank you for that question.
Another question we got in, has there been any analysis as far as particle size goes with the sediment that's being removed? So kind of discussing like kind of the influence of clay in soils compared to coarser textured sediments.
Mm. Yeah, that's a good question because as far as I know, the sediment that we use, it's technically not a soil, but it's a very high it's very high in organic matter. So that would be kind of like a histosol if it was on land. And that's very important because PCBs like to attach to organic materials. And actually, I remember when I was reading about the PCBs being found at the bottom of the ocean, a lot of times that's because the PCBs end up in fish and other marine wildlife, and then when they decompose, that sort of allows them to be deposited into the sediment as organic materials. And so, this is really important because uh, it shows that our biochar is still effective even in this really high organic material concentration sediment because the biochar itself is very high in organic matter. And so the fact that we can still see a significant decrease in PCBs going into the air with the biochar is really exciting. But we do plan to also conduct more extensive analyses on our sediment as part of our future experiments as well. Thank you for the question.
Yeah, we've gotten another or several other questions about the possibility of using LB400 as or for cleaning other chemicals out of um, water and things like that. So is there potential that it could help remove things like PFAS or heavy metals, other kind of toxins we see in the environment?
Yeah, I'm not totally sure about its effectiveness at degrading PFAS specifically, but I believe it has been used to degrade similar forever chemicals, similar to PCBs. And this method that we're developing with the biochar is also interesting because if we want to try to go after PFAS or other chemicals, we can potentially use different bacteria as well. And we can take our concept of growing it on or attaching it to the surface of the biochar and potentially also the encapsulation method that Chin developed to sort of customize the treatment based on the type of chemical we want to go after, and then also the environment. Because when I mentioned that we want to try to extract native bacteria from New Bedford, we could also, in the future, scientists could look for bacteria that could degrade PFAS under the specific conditions that are required at each contaminated site. And so that's one thing that really excites me about my research is that it can potentially be applied to, a wide range of different pollutants. And, that's kind of why, I got into this field, after studying soil science, is I specialized in soil microbiology at the time. And so I really wanted to be able to use what I had learned to help protect public health. And so, yeah, that's something that I'm really excited about. And thank you for the question.
Another question here. So would this method or does this method have potential for use for dredged material that's already in a containment facility and being dried out?
Yes, actually, the LB400 was first identified in one of these landfills that was filled with dredged material, and so that could be a way to help clean up a lot of these toxic dump sites because I've read a lot about these landfills potentially being hazardous, like if they leak out into the environment. And so, yes, it could absolutely be used to help clean up dredged material as well.
This is kind of going off the question a couple questions ago. Has there been any research into the effects of this bacteria on things like insecticides and neonics, pretty much? Yes, insecticides, things like that.
Unfortunately, I'm not aware of any research that used LB400 to go after neonics, um, but yeah, that's another interesting application of bacteria or what we call bioremediation. So just using bacteria to clean up harmful chemicals. Yeah, well, we do know that, PCBs are very similar to DDT, and so you could assume that LB400 would be able to degrade that. And then if the structure of the other types of pesticides is amenable to LB400, then it could definitely be used to degrade them.
Awesome. Well, that looks to be all of the questions we've got in. So again, thank you, David, for helping us out here today and teaching us a little bit about some of these chemicals and toxins out in the environment and cleaning those up. Again, for everybody watching, please tune in next week. We'll have Sabrina Ruiz with USDA. She's going to be talking about biomass and cover crops, kind of gaining some of those soil health indicators. So With that said, I'm gonna give the last words over to David if there's anything else you'd like to say.
I just want to say, thank you so much for the opportunity to present to you today. I really appreciate it. Another thing I'm really, passionate about is trying to take, my work, which is pretty complicated, and really share it, with the wider public. And so, it's really, it really motivates me that, normal people outside of academia are interested in my work. And so I, really appreciate you all for coming today. And it really helps, motivate me to keep going and doing this work.
Awesome. Well, thank you all for attending again. Tune in next week, and thank you, David, for a great presentation.
Thank you very much.
Yep.